Auzubillahi minash shaitanir rajim. Bismillahir rahmanir rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Today we are going to record the first year, chapter number 4, bioenergetics. So that is chapter number 4, bioenergetics. We are going to explain this. Now here we have used bioenergetics. Just look at here. Bio mean life, or we call them living organism. Life or living organism. And energetics term derived from energy. Energy means ability of a body to do work. That's called energy. So in simple definitions about bioenergetics, that is the study about transfer of energy in the body of living organism. That's called is bioenergetics. Chapter number 4, bioenergetics. Bio mean life or living organism, and energetics mean energy. So means the transfer of energy in the body of living organism is called bioenergetics. That's a simple definitions. While the main source of energy, that is the sunlight. So sunlight, that is considered to be the ultimate source of energy. Now this energy that has been captured by the autotroph. So autotroph, for example, plant. In these autotroph, they are fed by the herbivores. So herbivores feed upon the autotroph. Then carnivores, they feed upon the herbivores. So that's why energy is transferring from one organism to another organism. So we will discuss this energy which is related to the living organism. Or there is another definition as well. That is the study about qualitative and quantitative transfer of energy in the body of living organism. The study about qualitative and quantitative transfer of energy in the body of living organism. Here we have used the two terms. One is qualitative, another is quantitative. We should not compromise on quality. Why? The most purest form of energy that is present in the autotroph, in the fruits, in the vegetables. So we must be vegetarians. Why? Actually the diseases they caused by bacteria or viruses or some pathogens or parasites, they are host specific. So that's why we will be protected from this type of infection, contamination. And second, if we feed them these material for our food, so the top class energy is present in vegetables, mean fruits. And second, that's present in fish. Third, that's present in muttons. So then in broilers. So it's mean there is a sequence of energy. Some material they have a very large quantity, but quality that is zero. So that's why we have used this term qualitative and quantitative transfer of energy. Now here we are discussing the term metabolism. Metabolism means sum of all biochemical reaction. Sum of all biochemical reactions occurring in the body of living organism. Sum of all biochemical reaction occurring in the body of living organism, that's called metabolism. Metabolism is divided into two types. One is called anabolism and another is called catabolism. Now, anabolism is a constructive process, while catabolism is a destructive process. Anabolism, that is actually a process in which small and simple molecules combine together to form large and complex molecule. Here the energy is stored, because here the bond formation occur. The example is photosynthesis. So photosynthesis, that is considered to be the examples of anabolism, while catabolism, destructive process, that is the process in which large and complex molecules are break down into small and simple molecule with the release of energy. So here the bonds are breakdowns. So their example is respiration. So that's why we will first discuss the photosynthesis, then later on respiration, and different questions related to this topic. But before going to this topic, you must have an idea about this bioenergetics. So bioenergetics is the study about transfer of energy in the body of living organism, or the study about qualitative and quantitative transfer of energy in the body of living organism. That's also called as bioenergetics. So it means they are concerned with the energy. Here we are going to discuss about the photosynthesis. Photosynthesis, just again, that is actually consists of Greek word in which we use the term photo. So here the photo mean light, and synthesis mean manufacturing. Synthesis mean manufacturing. So what is photosynthesis? That is actually the process in which autotroph, for example plant, prepare their own food from inorganic compound that is carbon dioxide and water molecules, in the presence of sunlight and chlorophyll molecules. That is the definition of photosynthesis. Here we have used the term autotroph. Autotroph. So what's mean by autotroph? Auto mean self, and troph mean nourishment. Self-nourishments mean those living organisms which have the ability to prepare their own food. Those living organisms which have the ability to prepare their own food. For example, plant. For example, cyanobacteria. For example, autotrophic bacteria. So these are considered to be the different types of autotrophic organism. So accordingly to this reaction, accordingly to this definition, the reaction is given, that is Carbon dioxide and water molecules, they combine together to form glucose molecule with the release of oxygen in the presence of sunlight and chlorophyll. Now here we have write on the 6 water molecule, so that's why we have balanced the reaction. Whenever we write 12 water molecule, we must write 6 water molecule in the product. Now these two on the right side of reaction, that is called is reactants of a reaction, also called is raw material, and these are considered to be inorganic, while these two are considered to be the requirement of a reaction. These are the requirement of a reaction, and that one, that is considered to be the product of a reaction. Product of reaction, and that is considered to be organic and is called glucose, while this one, that is called is byproduct. That's considered to be byproduct, and that is oxygen and is inorganic in nature. So that is summarize reaction of the definitions which we have discussed. Now here we are going to discuss what is the role of carbon dioxide during photosynthesis process. Is carbon dioxide is required or not? Is it necessary? Yes, carbon dioxide is necessary for the process of photosynthesis. So we are going to discuss what is the role of carbon dioxide. Carbon dioxide, actually carbon dioxide fixation occur in dark cycle for what? For glucose production. For glucose production, say the carbon dioxide fixation occur. Now carbon dioxide, that is considered to be the oxidized. Carbon dioxide is considered to be the oxidized form of carbon. Carbon dioxide is considered to be the oxidized form of carbon. And carbon dioxide is important because carbon dioxide provide carbon and oxygen molecule to glucose. Why? Because actually carbon is the backbone of organic compound. That's why it's important. Why for organic compound? That is due to catenations. What is catenations? Catenation means self-linkage. Self-linkage. It means one carbon is linked with another carbon, so they have the ability to form a long chain. So that's properties only present in carbon atoms. That's why it's a key elements of organic compound. Just look at here. Now this carbon is present in the glucose. And what about this oxygen? Same, this oxygen is also present in the glucose molecule. So that's why carbon dioxide provides carbon and oxygen to the glucose molecule, or in a simple way that carbon dioxide fixation in dark cycle for glucose production, because glucose having carbon and it's organic in nature, so carbon element is considered to be the backbones of all organic compounds due to the phenomena of catenations. Second, now we are going to discuss then what is the role of water molecules. Here, water, that is considered to be the reducing agent. Water is considered to be the reducing agent. Now, what is their role? Their basic role is that they provide proton, mean hydrogen ion to glucose molecule. Here, just look at here. This hydrogen, we are observing that glucose molecule have hydrogen. So they get this hydrogen molecule from this water molecule. That's their basic function. Now, another most important things regarding to this role of water, that is CB1 Neal in 1930 proposed that oxygen in air comes from water, not from carbon dioxide. Look at here. They are talking about this oxygen. The oxygen which we are using during respiration. So this oxygen come from actually water. So this oxygens in the air that's come from the water, and that was first proposed by CB1 Neal. Later on in 1937, Robin Hill supported the idea of CB1 Neal by isolating chloroplast and use a molecule X instead of carbon dioxide. Still oxygen was released. How? Actually, this Robin Hill, he isolated the chloroplast. Chloroplast is a cell organelle and it's a double membranous. It consists of two membrane, one is outer membrane, another is inner membrane. This inner membrane consists of highly folded structures, we call them grana. And these grana are linked with one another through intergrana. Whatever, these are the different structures. Let's suppose we consider them that that's a chloroplast. So this Robin Hill used a molecule X, mean any type of molecule, but not the carbon dioxide. So that X is used instead of carbon dioxide. Now here there is no carbon dioxide in these reactions, and water molecule, means this X molecule is used to retain the hydrogen, and along with that reaction oxygen that was produced. So it's mean that the idea of CB1 Neal, that was a correct statement that he put forward, that the oxygen releasing from water, not from carbon dioxide. But science need more and more experiments, more and more evidences. So that's why later on another scientist, Samuel Robin, he performed an experiment in 1941, and he used the isotopes of oxygens. Isotopes of oxygen he used in an experiment. Isotopes are actually those having a same atomic number but different atomic mass. Oxygens have two isotopes. One is called as O16 and another is called O18. That's an oxygen, and one is called O16 and another is called O18. Now, what he actually done for his experiment, he took two plants growing in a pot. One is A and another is B. Then these plants, all the conditions they are provided, that is for required for the photosynthesis process. Then he cover these two pot with a covering. Let's suppose that sun is present and sunlights are coming, because we have discussed that sunlight's chlorophyll, these are the requirements. Same case also here, sun is present and their lights falls on the plant. Now what did he done? He provide carbon dioxides and water molecule in a controlled way, and he use isotopes O18s of water and O16 of carbon dioxide. Now the process of photosynthesis started because sunlight is present, carbon dioxide is present, water is present, all the materials are present. So then photosynthesis process been started. During photosynthesis process, plant release oxygen molecule, and this oxygen molecule that was collected by a scientist, he observed that this oxygen that was released during this process, that was all of them of O18. So what does it mean? That has been cleared that oxygens come from the water, not from the carbon dioxide. In next experiment, he used the O16 isotopes, and of carbon dioxide he used O18. Light is there, carbon dioxide is there, and water molecule is there. So chlorophyll is present and the plant have started the photosynthesis process. Then he again collect the water oxygen molecule from the photosynthesis process during this process, and that was observed O16. So O16 was actually coming from the water. So hence it's proved that oxygen come from water, not from carbon dioxide. So during experiment A, we studied that water molecule and carbon dioxide, they combine together and produce glucose molecules. So this glucose molecule that is considered to be organic in nature. That is considered to be organic in nature. In experiment first, he used the isotopes O18 of water and 16 of carbon dioxide. So here the oxygen that was coming. And the next experiment, he then used O16 of water and O18 of carbon dioxide. So here they have collected the O16 oxygen molecule. So hence proved that oxygen come from water, not from carbon dioxide. Now we have another general type of idea related to this topic, that is photoautotrophic bacteria. Photoautotrophic bacteria. So photoautotrophic bacteria, there also photosynthesis take place, but they use hydrogen sulfide instead of water, and they are releasing sulfur. If photoautotrophic plants use hydrogen sulfide instead of water in releasing oxygen and releasing sulfur instead of oxygen, same means that the plant, they are actually using water molecules, and obviously they will release oxygen because the sulfur of hydrogen sulfide is released as a byproduct. So definitely oxygen of water will be released as a byproduct. That's another statement that we should recommend for this experiment as well. And along with this, we can also calculate about the action spectrum. Action spectrums. Action spectrum that can be calculated by collecting the amount of oxygen produced during photosynthesis. That one, this oxygen that is producing during this process, we collect them. What's mean by X-ray spectrum? The light that falls on the plant body, they absorb them, they trap them. Then during photosynthesis process, light reaction in dark cycles to convert this light energy into chemical energy. So that's called action spectrum. So we calculate the action spectrum by collecting the amount of oxygen. If greater the amount of oxygen, so the action spectrum will be greater. If the oxygen concentration, if oxygen product is less, so action spectrum will be in a small, low quantity. So that's why that's directly related to them. Now, then after we are going to discuss then what is the role of chlorophyll molecule during the process of photosynthesis. So because we have discussed the role of carbon dioxide, then we have discussed the role of water molecule, then we are going to discuss what is the role of chlorophyll molecule. So role of chlorophyll molecules. What is a chlorophyll? Chlorophyll is actually organic pigment. That is actually organic pigment, and that is used to absorb sunlight. Their main function is to absorb sunlight. That's their main function. But keep in your minds that there are there are a lot of chlorophylls, but actually we are mainly focusing on two types. One is called chlorophyll A and another is called as chlorophyll B. If someone asks from you what's the formula of chlorophyll A, so their formula is C55H72O5N4Mg. And chlorophyll B formula is C55H70O6N4Mg. Now again, chlorophyll A, that is considered to be the most abundant or most common type of chlorophyll. And second, chlorophyll A have a methyl group as the functional. So they have functional group, that is their methyl. While chlorophyll B, they have a carbonyl. Carbonyls mean they have a aldehydes. They have aldehyde as a functional group. So that's why sometimes we say about the chlorosis. So chlorosis term, why we use them? Chlorosis term is basically used when there is a yellowing of the plant leaf. Why the plant leaf yellowing occur? If there is a deficiency of nitrogen or there is a deficiency of magnesium, so the plants are unable to produce the chlorophyll molecules. So here we are going to discuss about the chlorophyll structure as well. And second thing, one thing more, that the chlorophyll A, they have a bluish green color. They are bluish green, while the chlorophyll B, that is a yellowish green. That is a yellowish green. Now, chlorophyll molecules, if you discuss the structures of chlorophyll molecules, let's suppose chlorophyll molecule, they are consist of two parts. One is called head and another is called tail. Head, we call them porphyrin, while tail is called is phytols. Tail is called phytol. Porphyrins, that is considered to be hydrophilic in nature, and phytol, that is considered to be hydrophobic in nature. Second thing, porphyrins, they are made up of 4 pyrrole ring having magnesium ion in its center. While this phytol, which is hydrophobic in nature, phytols, their formula, that is C20H39s. By nature, phytol is diaterpenes. That is diaterpene. Why? Actually, one is called as monoterpenes. So monoterpene, another is called as a diterpenes. Monoterpene is actually made up of two isoprenyte, while diterpene is made up of a 4 isoprenyte unit. While one isoprenyte, a single isoprenyte consists of 5 carbon atoms. If a single isoprenyte has 5 carbon atoms, so there are 4 isoprene. So 4 multiplied by 5 is equal to 20. So that's why they have 20 carbons along with 39 hydrogen atoms. So that's a diterpene. There is another tail is also present, but that's not a very small in few elements combined together. Phytols, that's made up of diterpene, and that phytol, that is considered to be the embedded in thylakoids. This part is embedded in thylakoids, which made the grana, and grana then interconnected with the intrograna. Another thing that is in case of chlorophyll A or chlorophyll B, their functional groups, they are attached to second pyroles on carbon number 3rd. Let's suppose that is a magnesium ion, and that is considered to be the 1 pyrrole ring, and that is considered to be the second pyrrole ring, although their formula is a very difficult. So these are the different pyrrole rings. So there are total 4 pyrrole rings are mentioned. These pyrrole rings, they are also linked with one another as well. They are also linked with one another as well. That's the first pyrrole, second pyrrole, third pyrrole, and fourth pyrrole. Now, to the second pyrroles, on carbon number 3rd, methyl group is attached in case of chlorophyll A, while in case of chlorophyll B, then they have a aldehyde functional group is attached. So that's the difference between chlorophyll A and chlorophyll B. So that's all about the chlorophyll molecule. Now, then we are going to discuss about the sunlight. So chlorophyll, they absorb various wavelength color light. So we will discuss with the role of sunlight about this wavelength of chlorophyll molecule. Now we are going to discuss then what is the role of sunlight. Keep in your mind that sun, that's the ultimate source of energy. 40% light coming from the sun reach to the earth's surface, 10% falls on the plant body, and only 1% plants use them for their energy resources. Only 1%. Now, sunlight actually, these consist of 2 parts. One is called as visible, and another is called as invisible. So this visible range in between gamma and radio rays. So as if this visible range in between the gamma and radio rays. Invisibles before the ultraviolet rays, and after the infrared. These are called the invisible light. We are mainly focusing on the visible color lights, that's in the form of waveguides. Violet, V stand for violet, I stand for indigo, B stand for blue, G for green, Y for yellow, O for orange, and R for red. So there are 7 different color lights is there. And one thing most important, that the blue color light is a most suitable color light for photosynthesis, blue and red. But among them, blue color light is the most important color light for photosynthesis. While the green color light, that is being reflected. So that's why we consider that is the least important color light for the photosynthesis. Now this color lights, visible color lights, most probably range from 380 to 760 nanometers. That is the visible color light. Visible color light range from 380 to 760. Yes, there may be a 10 to 30 plus minus occur in the value, because we are going to take the average value. Now from 380 nanometer to 430 nanometer, this color light is considered to be the blue color light, while from 500 to 600 nanometer color light, this color light is considered to be the yellow color light. Then from 670 to 730 nanometer color light, and that's considered to be the red color light. Now this blue color light, that has been absorbed by the chlorophyll A molecules, while 500 to 600, that is being absorbed by the carotenoid, while 670 to 730 nanometer, that is being absorbed by the chlorophyll B molecules. So these different wavelength color light are absorbed by the different photosynthetic pigment. Now that's why we call them absorption spectrums. We call them absorption spectrum. So this absorption spectrum can be used to calculate the spectrophotometer. Spectrophotometer. So spectrophotometer, that's an instrument. That's an instrument, that's a device through which we can measure the different wavelength absorbed by the different photosynthetic pigment that we have mentioned here, different wavelength color light. So that's why this sunlight is the most important, because we have discussed that the main source or ultimate source of energy. That's why it's most important. Now another thing very important, that is this sunlight, that is being trapped by the plants and converted into chemical energy, in the form of glucose molecule C6H12O6. Who have the ability? Autotroph have the ability. Autotroph have the ability to convert them. Now autotrophs, these are the, actually they are talking about the plant. They are talking about the cyanobacteria. They are talking about the photoautotrophic bacteria. Photoautotrophic bacteria. So they have the ability to convert this sunlight into chemical energy. Why? Sunlight, that's in the form of photon. That is in the form of electromagnetic wave. That is in the form of discretes. That's in the form of radiant energy. So sunlights are, that's a light energy. That's a light energy. So sunlight, that's a light energy, that's a radiant energy, that's a photon, that's electromagnetic wave, or discretes are considered to be the packet or bundle of energy. Packet or bundle of energy. Packet or bundle of energy. So plants convert them into the chemical energy, then they are used. Now another thing we are going to discuss here, What about accessory pigments? These are the additional pigments required during photosynthesis. Accessory pigment, that is about carotenoids. As we have discussed that the carotenoid absorb 500 to 600 nanometer color light, and that's considered to the yellow color light. And their wavelength 5 to 600 further consists of one having carotene and another having xanthophyll. And they are actually, they have a yellow color. Carotene have the ability to absorb red and orange color light. So red and orange color light that is being absorbed by the carotene, while xanthophyll absorb yellow or brown color light. So that's why sometimes we say that the carotene absorb red, orange, yellow or brown color light that has been absorbed by the chlorophyll. And this carotenoid absorb light, then it passes to the chlorophyll A molecules. This passes the light energy into the chlorophyll A molecule. Another most important thing, that is Einstein, he put forward a theory, photoelectric theory. We call them photoelectric theory. Accordingly to this theory, one photon excites only one electron. Einstein put forward a theory that's called as a photoelectric theory. Accordingly to photoelectric theory, one photon excites one electron. One photon excites one electrons. So mean that sunlight there beams fall on the plant body, they have the ability to excite the electron. Now here we have discussed the reaction that the carbon dioxides and water molecules, they combine together to form glucose molecule whose formula is C6H12O6 along with the release of oxygen molecules and occur in the presence of sunlight and chlorophyll molecule. It's not easy. That's we have write down, the reaction will be done so quick. No. Let's suppose, consider that is a plant growing in a pot, and they have their different body parts. They have their root, they have their stem, they have their leaf, they have different parts. And here the sunlight is present, and we have discussed that 40% of light there comes toward the earth's surface, 10% falls on the plant body, and only 1% plant use them during the process of photosynthesis. So these are the leaf. The leaf have they actually take water molecule from the soil, and so water is being reached to the leaf surface, and they absorb carbon dioxide from the air through the stomata, and then they combine them. But there, by this process, they release oxygen molecule. This oxygen molecule, they use them in their own respiration as well. But there is one point, we call them compensation points. This point is called compensate point, where the rate of oxygen become equal to the rate of carbon dioxide. Now, if we take the leaf from this plant, we know that the leaf, their part that's called the petiole, and this part is called the lamina, and they have a central vein that's called as a midrib, and they have a small other veins are present. Actually, these are the various vascular bundle. When we take the transverse section of the leaf, by taking the transverse section of a leaf, we will observe under the microscope the different mesophyll cell. One thing more, A single mesophyll cells contain 20 to 120 chloroplast. And one chloroplast contain 10 to 100 grana, and one grana contain about 50 thylakoid piled. Now here, and second thing, mesophyll cells are of two types. One is called a spongy mesophyll cell, and another is called a palisade mesophyll cell. Palisade mesophyll cells are properly arranged as compared to spongy, because spongy mesophyll cells are dispersed. The most probably the photosynthesis taking place in the palisade mesophyll cells. Let's suppose we should consider that is a single chloroplast. And we have discussed that the chloroplast, their diameter is 4 to 6 micrometer and is a double membranous. The inner membranes, they forms folded structures which we call them grana, which is linked together with a intragrana. So this grana part, that is a green due to the presence of chlorophyll molecules. And we have find out a cluster is there. We call them photosystem 1 because it was first discovered. Later on we have discovered another cluster, that's we call the photosystem 2. So simply one is discovered first and second is called later on. That's why we call photosystem 1 and photosystem 2. These two photosystems are connected with electron transport chain, which we call them, that is considered to be the light reactions. After that, dark cycles occurred. So means this glucose production is related with the light reaction and with the dark cycle. So first we will discuss the light reaction, then we will discuss the dark cycle. So first we are going to discuss the detail about light reactions. Light reaction is also called light dependent reaction. Light reaction is also called light dependent reaction. It's also called Hell reactions. It's also called photosynthetic electron transfer reactions. Light reaction, it's also called light dependent reaction, it's also called Hell reaction, it's also called photosynthetic electron transfer reactions, and also called as photochemical reactions. This light reaction that's actually occurred on grana. Light reactions need sunlight. If sunlight is there, so light reaction will be occurred. If there are no sunlight, there will be no light reaction. And this light reaction that was discovered by Robin Hill and Bendall in 1960. In some book they have write down 1961. And yeah, they are discovered by them. Now light reaction is, if you are going to discuss about the end product, if you are going to discuss about the end product of light reaction, so the end product is consist of 3 ATP, 2 NADPH2 and 1 oxygen molecules. That is the end product of light reactions. And light reactions, two types. One is called as non-cyclic light reaction, and another is called as cyclic light reactions. One is called as non-cyclic light reaction, it's called cyclic light reaction. In non-cyclic light reactions, ATP NADPH2 and oxygen molecule production occur, while in cyclic light reaction ATP production take place. First we will discuss the light reaction, then we will discuss the non-cyclic, then we will discuss the cyclic light reaction. Let's suppose we have discussed that there are two types of photosystem. One is called photosystem 1, another is called photosystem 2. As I have told you that one photosystem 1 that was first discovered, then photosystem 2 was reported. So that's why their names are based upon their discovery. Although these photosystems are different in their structures based upon the number of chlorophyll molecules and carotenoids molecules, about their thickness and about their if they are thin, and so many other factors about their location, but on the basis of wavelength they are also different from one another as well, because of presence of the chlorophyll's molecule or photosynthetic pigment, their wavelength also change. So let's suppose these are the two photosystems. That one is called as photosystem 1. Range of their light is 700 nanometer. That's photosystem 2, and range is 680 nanometers. Each photosystem, if it's photosystem 1 or it's photosystem 2, they are made up of antenna complex. That's an antenna complex. This antenna complex is made up of one carotenoid and another chlorophyll B molecule is present. So this antenna complex have carotenoids and chlorophyll B, while the reaction center they have the chlorophyll A molecules. So where does the chlorophyll A molecule is present? That's present in antenna in the reaction center. So that is the reaction center where the chlorophyll A molecule is present. Now, sunlight, it's in the form of light energy, radiant or photons. These lights falls on the plant body, and first they excite electron from photosystem 1. So here when the electron excited, so the electron that has been received by the phosphates. Phosphatine is considered to be the primary electron acceptors. They are the primary electron acceptor. So this electrons jump from chlorophyll A to phosphatines. That's the primary electron acceptor. Then these electrons from phosphatine that is being received by the ferredoxine reducing substance, and that is a iron containing protein molecules. It's an iron containing protein molecule. So these two electrons is being received by the ferredoxine reducing substance. From ferredoxine reducing substance, these electron that is being received by the NADP. So this NADP then take these electron and will form NADPH2. So that's why NADPH2 is considered to be the final electron acceptor. Now the deficiency of electron of photosystem 1 that is being covered by photosystem 2. How? Same light. Now it's falls on the photosystem 2. Same procedures. Phosphatine, which we call them primary electron acceptors. So the light falls on the chlorophyll A, that will be excite the electron which is received by the phosphatine. From phosphatine, this electron that is being received by the quinones. From quinone, these 2 electrons that is received by the plastoquinones. From plastoquinone, these electrons are received by the cytochrome 553. Simply we call them cytochrome B. From cytochrome 553 that has been received by the cytochrome 559, we call them cytochrome F. Then it has been received by the plastocyanine, and plastocyanine is a copper containing protein, and this electron has been delivered to the photosystem 1. So through this way the electron passed. That's why we call them electron transport chain. Now when electron move from plastoquinones to cytochrome B, here the ATP production take place. When it's moved from 553 to 559 B to C, another electron production take place. So that's why these ATP production take place. This type of ATP production we call them photophosphorylations. The process of formation of ATP during light friction is called photophosphorylation. Now there is a deficiency of electron in photosystem 2. What happened during this process? There is a reactions take place. The name of this process called is photolysis. Photolysis. Photo mean light and lysis means splitting. Now here the water molecule that was used in the photosynthesis process, that has been split into 4-hydroxyline and 4-hydrogenines. Then 4 hydroxyl combine to form 2 water molecules, oxygen and 2 electron. Now these protons, hydrogen ion that is being received by the NADP. So that's why NADPH2 production take place. While oxygen that we have discussed that has been released as a byproduct, because if you people remember, we have discussed that the carbon dioxide and water molecule, they combine together to form glucose molecule with the release of oxygen. Now during this light reaction we have studied the water. We have studied the water, what's their role, where they participate. We have discussed the chlorophyll molecules. We have discussed the carotenoids molecules. Now that's considered to be the non-cyclic light reactions. The final electron acceptor during this non-cyclic light reaction, that is a NADP. And NADP is a part of photosystem 1. While plastoquinone or quinone is a part of photosystem 2. The 700 nanometer observed by the photosystem 1 and 680 observed by photosystem 2. Photolysis occur in the photosystem 2. So there are different questions they may ask. That's a non-cyclic. If we discuss the cyclic light reaction, so cyclic light reactions, this process, during this process, this photosystem 2 that has been blocked. When photosystem 2 is blocked, so only cyclic light reactions will occur. Now that's a cyclic light reaction. For this case, the final electron acceptor, that is photosystem 1 by itself, because electron passes from ferredoxine reducing substance come back to the photosystem 1. So that's why final electron acceptor is photosystem 1 by itself. So light reactions take place in the presence of sunlight. If sunlight is present, so light reaction will be there. If there is no sunlight, there will be no light reaction. But second one, which we call them dark cycle needs the product of light reactions. So next topic, that is about the dark cycle, which is also called is light independent reactions. Dark cycle is also called the light independent reaction. That's also called as carbon fixation cycle. That's also called as Kelvin cycle. That's also called as C3 cycle. That's also called as thermochemical reactions. That's also called as reproductive pentose phosphate pathway. That's also called as carbon dioxide fixation cycle. Now, dark cycle, during dark cycles, glucose production takes place. Glucose molecule is being produced. Where does the dark cycle occur? That's occur in the stroma of a chloroplast. What does they need? They actually need the end product. They need the end product of light reactions. What is the end product of light reaction? The end product is ATP and NADPH2. So they need this. And who discovered dark cycle? That was discovered by Kelvin, Melvins, and Beauchamp in 1950. So in 1950, Melvin, Kelvin, and Beauchamp discovered this dark cycles, and they received Nobel Prize in 1961. What is a dark cycle? And dark cycle consists of 3 steps. In first phase, carbon dioxide fixation occur. In second phase, NADH2 reduction take place. In a third phase, regeneration of RUBP take place. Now coming toward this cycle, let's suppose there is a molecule, we call them RUBP. RUBP stand for 1,5 ribulose bisphosphate. Ribulose bisphosphate. What is the nature of RUBP? RUBP is a carbohydrates. In carbohydrates, they are monosaccharide. In monosaccharide, that's a pentose sugar. Means they are contain 5 carbon atoms, and they have a ketone as a functional group. They have a ketone as a functional group. So RUBP, 5 carbons compound. There are 3 molecules present, and they receive carbon dioxide molecule in the presence of enzyme called Robesco. Here the Robesco enzyme acts as a carboxylase. They act as a carboxylase. They will form 6 carbon compound, 3 molecules are formed, and that's unstable, so that's why they have no name. Then this 6 carbon compound break down into 3 carbons compounds, 6 molecules are formed. That's why these molecules, that's called PGA. So PGA means phosphoglyceric acids. So phosphoglyceric acid or PGA, the first stable compound of dark cycle. Then this PGA is converted into PGAL, phosphoglyceraldehyde. During this process, now 6 NADPH2 that is being consumed, and as well as 6 ATP molecule is also utilized. So that's why that's a second phase. First phase was the fixation of carbon dioxide. Second phase, the reduction of NADPH2. Now what happened during this process? Out of 6 PGL molecules, now one molecule comes out from this cycles. Then how many left it? 5. Total 6 PGL molecule is there. In this 6 PGL molecule, 1 PGL molecules comes out from this cycle. That's in our first round. 1 PGL molecules comes out. How many left behind? Total 5 PGL molecule left behind. Each consists of 3 carbons. Now, 3 plus 3 combined together to form 6 carbon compound. 6 plus 3 combined together to form 9 carbons compound. So 9 carbon compound split into 5 carbon compound and 4 carbon compound. Now 5 carbon compound, actually that's RUPP. Then 4 plus 3, 7 carbon compound, and 7 plus 3, 10 carbons compound. 10 carbons compound split to form 5 carbons compound and 5 carbon compound. 5 carbon compound is RUPP. So it means RUBP molecules are regenerated. With the regeneration of each RUBP, 3 ATP is also consumed. So during this cycle, total 6 ATP is consumed. So that's why one dark cycle consume 9 ATP and 6 NADPH2. Now again this cycle we start. RUBP, 3 RUBP with 3 carbon dioxides form 6 carbon compounds, change into 3 carbons compound, then which is called PGA. It's a first stable compound, that's why we call them a C3 plant. PGA is changed into 6 PGL. Again 6 NADPH2 and 6 ATP are consumed. Out of 6 PGL, another PGL molecules comes out. That is now the second round. Second round, another PGL molecules comes out. Then these 2 PGL molecules, they combine together and they will form a glucose molecule C6H12O6. So then remaining 5 they rearrange to form RUBP. So that's why the 2 dark cycle consumed 18 ATP and 12 NADPH2. So that's why one glucose molecules need 18 ATP and 12 NADPH2 for its productions. So that's about the dark cycle. So that's why glucose molecule has been synthesized. And we have discussed about this reaction, that's H2O and carbon dioxide. Carbon dioxides and water molecules, they combine together to form glucose molecules with the release of oxygen. So we have observed that this carbon dioxide that is being utilized here in dark cycle. And we have discussed in light reactions that water molecule was used over there, and we have observed that glucose molecule has been synthesized, and we have observed that oxygen was released during light reaction. So that's why that is called as a the production of glucose molecule need a 2 dark cycles. So that sometimes this process actually takes place in the daytime because it's need the end product of light frictions. So if the light is there, so light friction will be there. If the light friction, so their product will be there. So that's why dark cycle will be proceeds. So we are going to wind up our lecture over here, and inshallah we'll meet together in next lecture. So wish you best of luck. Allah Hafiz.